So when we have several amino acids coming together to form a peptide, we can form an alpha helix. An alpha helix is an example of a secondary structure of peptides. So remember, we have primary, secondary, tertiary, and quaternary peptides. Primary refers to that primary amino acid sequence, just the sequence. Now, secondary refers to the actual folding in a way. So what happens is that, remember that our double bond with our oxygen, so we have our alpha carbon over here, and we have that double bond on our oxygen, and we have our peptide bond right over here. Now, recall that this peptide bond actually has a partial double bond character because these electrons over here shared between the carbon and the oxygen are delocalized. So nitrogen is actually able to form a double bond with carbon. This gives the peptide bond a double bond character. This double bond character generates that positive charge on the nitrogen and that negative charge on that oxygen. And this is very important because these charges, which we can see right over here, can be used to form hydrogen bonds within our alpha helix. And we'll take a look at that in a second. But this double bond character also restricts rotation about this carbon. The only rotation we see is about the alpha carbon. We do not see rotation over here, and we can see that in this diagram. In these blue boxes, this is where the peptide bond is depicted, and these will not rotate, but our alpha carbon is free to rotate. Now, we have a diagram of our alpha helix, and we can see it's similar to the alpha helix that we see in DNA. So this is a single strand, and our R groups, so remember that amino acids have R groups, which are going to point outwards. Now, these R groups, if these R groups are small, they're actually not going to prefer the alpha helix structure. They're actually going to prefer the beta sheet structure. Beta sheet is another example of a secondary structure. So if the R groups are small, it's going to prefer the beta sheet. But if our R groups are too big, we can imagine that'd be too much crowding, and that would actually generate some repulsion. So we don't want too many large, bulky groups, and we don't want small groups either. So that's an important feature of an alpha helix to occur. So the R groups are going to point outwards, and the inner part of this alpha helix is essentially the backbone bonding via this peptide bond. So if we take a look over here, we may have, for example, a nitrogen which has a hydrogen, and up top over here we may have an oxygen, and we'll be able to form some hydrogen bonding, because remember that this nitrogen actually has a partial positive charge, and this oxygen actually has a partial negative charge because of the delocalization of the electrons. We can draw a better diagram, and we can actually say something like this. So we have our oxygen, which is negative, this is positive, and it's delocalized. So this would actually be partial negative and partial positive, and those electrons are delocalized. So since we have that partial negative and partial positive charge, we can see that hydrogen bonding on the backbone. And that hydrogen bonding, it's important to remember that this hydrogen bonding is not between our R groups. The alpha helix structure is not related to how R groups interact with one another. R group interaction is related to tertiary and quaternary structures, not our secondary structures of an alpha helix or a beta sheet. So in our alpha helix, our nitrogen and oxygen are forming hydrogen bonds, and our R groups are pointing on the outside. It's also important to note of the trans configuration of our peptide bond. Our oxygen points up and our hydrogen points down on our nitrogen. This trans configuration is very important. Now, it's another important thing to know of is that this hydrogen bonding, it's not exhibited between neighboring peptide bonds. This is actually depicted amongst every 4 amino acids. So every 4 amino acids, we see a hydrogen bonding where our nitrogen and oxygen are able to interact via hydrogen bonding. And that's mainly all we need to know about alpha helix. Remember that it's a secondary structure. So it's an interaction via hydrogen bonding between our oxygen and nitrogen. Our peptide bond has a partial double bond character, which restricts rotation, but we do see rotation about our alpha carbon. We have that trans configuration, and our R groups are also pointing outwards. And remember, we do not want small R groups because they will actually prefer beta sheet, and too many big bulky groups are going to create some steric hindrance. And that is essentially the alpha helix structure of peptides.